do I get to introduce Tal? Okay. Hey everybody. Hope everybody's having a good start to the week and that the eclipse didn't cause any major problems. So just real quickly, Tal is our speaker today. I really appreciate it. He uses the intersection of various disciplines, computer science, biology, physics, etc., to understand the antibody responses following vaccination. He got his PhD in biophysics at Caltech. During his postdoc, he shifted lanes into immunology, where he joined Dr. Bloom's lab at the Fred Hutch Center, where he studied antibody response to influenza. He also did a year-long sabbatical as a software developer, as well as what we'll hear about today. His research uses intersection of these various disciplines to understand antibody responses. He comes from the La Jolla Institute down the road. So I'm going to let you take it away, Tal.
Thanks, Davey.
Oh, yeah, there's our next seminars. Maybe we should go back to that. But we'll do it at the end. It's okay. Go for it. Thanks.
All right, so I'll share my slides then. Yeah. Okay, assuming everybody can see the cursor and everything. So my name is Tal Einav. I'm delighted to be here, and my lab is formed on the opinion that nowadays we can do anything once we have enough data. It's really the era of data, and what is possible is really limited by how far we're willing to push that. And in the case of what the Prepare Institute does, that translates into questions such as how can we use your current immune state to predict what happens after vaccination? We focus a lot on the antibody response at the moment. And so what does your antibody response look like now? And what will it look like if we give you some kind of immune perturbation? We study viruses such as influenza, HIV, SARS-2, things that rapidly evolve, and cases where we're starting to get treatments or vaccines or some way to control the virus. And specifically in each of these cases, but especially for flu, which is what I'll talk about today, the antibody response is highly polyclonal. So we don't just make one antibody against it, we make hundreds. And the whole process is constantly evolving. So the antibody response within us, each time that we're exposed or vaccinated or infected with a new virus, it shifts along with that virus, kind of this race mentality that keeps moving on and on. And if we are to roll back the clock just a little bit, then we can ask two key questions here. The first is, what happens on the virus side? What will be the next variant that emerges up top? And the second is, even if we knew exactly what was going to happen, is that good enough in order to make a good vaccine? And the answer with flu turns out to be a very emphatic no at the moment, because most seasons we do indeed guess correctly on which variants will emerge, and yet flu vaccine effectiveness is still only 20 to 50% on average. Which begs the question of what exactly does it take in order to understand these specific viruses well enough in order to predict not just how they will act in the wild and which ones will dominate, but also what will happen when we put them into a vaccine form. What response will they elicit within us? And so my lab studies the complementary question on the bottom half of the slide, which asks, if we knew what was going to happen, can we predict what will happen whenever we put it into a vaccine form? What will be the antibody response if we give people one vaccine over another? All right, so that's the stage for where I want to go. I'll just give you a sense of what the landscape looks like with the data sets that we have now for flu, which is probably one of the best studied viruses of all time. Just focusing on the H3N2 vaccine strains that we get every year, the H3N2 virus was the same in 2016-17, and then it changed. And for the past 2 years, we had the same H3N2 vaccine strain once again. It'll change in 2024. And so for each of these years, we have at least one large-scale study, sometimes multiple, that takes people who were given that specific vaccine, measures their antibody response, so takes a drop of their blood, looks at their serum response, and asks how well does it inhibit different influenza variants. And the variants that are typically examined are these historical variants, and people love especially the vaccine strains. So they'll pull these specific viruses, as well as 10 to 20 other ones, and they'll measure what's the response pre and post vaccination from these individuals, typically looking at around 1 month post-vaccination, sometimes going out to 3 months, occasionally 6 months, 1 year to see the long-term response. And so we have these types of data sets for pretty much every year that you can imagine, going back at least a decade. And I'll focus today specifically on these hemagglutination data, which means how well does the antibody response prevent the virus from agglutinating red blood cells. So something halfway between binding and neutralization. And we'll have these types of data sets for every year. So in 2022, for example, we'll have our own set of, in this case, 4 different large-scale studies that measure the pre and the post-vaccination response. And our goal is to learn the rules from one of these scenarios and then use that to predict what happens in a future year. Using models such as decision trees going all the way up to neural nets, looking at everything from not just how the antibody response looks like against variants, but even now starting to look at infection history, age, genetics, and other factors like that. So you can see that this isn't a perfect prediction challenge, if you will. I mean, the variants that are examined are constantly changing over time. So new ones will pop up in some years, old ones will go away. But there's still enough overlap between these data sets that we should be able to ask, well, if we know how this orange variant behaves up here, can we predict how it behaves down here in the next data set that we have? And on and on and on for every virus that we can that overlaps between them. And of course, sometimes we have this difficult scenario of the vaccine changes. In other years, we get very lucky. And in 2022, 2023, the vaccine didn't change at all for the H3N2 component. So in this case, last year, when everyone got their flu vaccine, I personally think that there was no reason not to immediately ask, well, let's just blind ourselves to what happened here and see if we can predict it based on all the data that we have. Can we know who was going to respond weakly, who would respond strongly, and why? And so this is the name of the game. It's what we do. We look at the roughly 100,000 different vaccine responses that we have measured for the past decade, and we ask, how can we incorporate all of those data together to get these universal relationships that will help us predict the responses? All right, so that is the 30,000-foot overview of what we do. And I want to tell you about the actual prediction mechanisms that we're using, how we're thinking about this, what the results look like. I'll mention just a little bit about where we're going with this in terms of differentiating between weak and strong responses. Who are these strong responders and what are their characteristics? And then I'll mention finally what I'm doing here with the UCSD Prepare Institute and the project that I currently pitched about making these personalized vaccine recommendations for influenza based on the vaccines that we already have ready here from the past few years. Okay, so starting with the prediction framework. So going back, these are the vaccines that we have looked at in roughly the past 8 years of influenza. You can imagine that there are different levels of complexity in terms of what you want to predict. You can start off by doing the most straightforward prediction challenge, which is that you take the different vaccine studies that we have from the same year. Like, let's say we have 4 studies that were done in 2022, and you can ask, can one of those studies predict the other 3? And then can the second study predict the others? And can the third predict the others as well? So this should be, in a sense, the easiest prediction challenge that you can make. You can then take one step up and you can ask, what happens if we predict across years that had the same influenza vaccine? And finally, you can ask what happens between various years. It doesn't matter if the vaccine stayed the same or not, but how many ways can you just predict across different data sets? And what I'm showing here, these numbers, they are a new unit of measurement that I'm creating here, which is a data set prediction. So not just predicting 150 values, but rather 150 cases where we go from one study to another study, with these studies typically having something like 1,000 measurements. So 150,000 values is what we're predicting here. And that gives us a very robust way to know, is this prediction framework good or is it not good? Because we don't just predict a few values, we're predicting a whole myriad of different responses here in each of these cases. So we get to sift through these 150 ways that we can predict from one of these studies that we have to another one. We can ask, when does it break down? For example, if you take this old 2017 study, can it predict the responses in 2018? And can it do 2019, 2020? At some point, you expect that you stop being able to predict because you're trying to predict from something happening in 2017 all the way out to something far into the future. How far does that crosstalk work? And when are you just comparing apples and oranges and it no longer helps you out?
I realized I got so caught up in stuff last week before, and I was only late in the week. I learned Roberta was going to be away this week.
Yeah.
But you might be the person.
Okay, so the question becomes, how do you predict between different data sets in this scenario? And what I'm showing you here is still only pairwise prediction. So you're taking one vaccine study and predicting another vaccine study, but there's no reason why you can't take clumps of vaccine studies and train on that and use it to predict a new data set. And once you do this, the number of combinations here is just combinatorially increasing. We have 65,000 different ways to take different subsets of the data sets that we have here and predict the remaining studies. And so at this point, this is why I say that we're really no longer data set limited here. The number of ways that we can make predictions is enormous, and we can sift through this vast landscape and ask which ones give us the proper information. How far back in time should we pull information, and which vaccine studies inform other vaccine studies in order to make these robust predictions? Okay, so I want to show you what our predictions actually look like. How well do they work? I'm going to skip, just for expediency, this point number 1, studies in the same year, skip right to point number 2, and look at studies that predict across consecutive years when the vaccine strain does not change. So going back here to 2016 to 2017, we're going to pull one vaccine study from 2016, learn how the features behave in there, and predict what happens in 2017 when the vaccine did not change. So this is pictorially what the setup looks like. We have hundreds of different individuals, each of them measured against dozens of different viruses. I'm just showing a little subset here. We learn what happens to each of these viruses post-vaccination. So for the green strain, we learn how this value up here is based off of all of the different characteristics back here. So how did a person respond to the blue, teal, green viruses here? And how does that predict this particular value up here? And then we do that for the second row and for the third row. And we use the pre-vaccination only data that we have from 2017, apply the algorithms that we learn, and try to predict a priori what will happen in 2017 and compare those predictions to the measurements. So here's what the results look like for us. I'm just showing you 4 individuals in 2017, their pre-vaccination landscape. So this is what happens at day 0, right before they get the vaccine. We measure their antibody responses against many different influenza variants here on the X. And you can see, for example, for this person, they have a moderate response against this vaccine strain before vaccination, and then it goes down for an older strain, back up, back down, to a very weak response down here, and then maybe a slight bump once you get to these very old viruses. And these other people, they look similar-ish. Some of them can have a higher response, some of them can have a just fundamentally different response. And in a normal talk at this point, I would just show you, here's what the post-vaccination data looks like, and we talk about it, and I'd point out a few features, and we move on. But I think that's a missed opportunity, because I look at hundreds of these antibody landscapes each and every day. I should know by now, I should have a gut instinct on what do I expect should happen. And so this is the way I want to give now every single talk for the rest of my career, is before showing results, I want to give you my impression of what they should look like. So here it is for this top leftmost panel, I think that when you give somebody this vaccine strain at the very right, that it'll bump up their response up there. And then there'll be diminishing effects. As you get to these older and older viruses, you'll see a smaller and smaller response. And then the curve basically hugs the pre-vaccination landscape. And I don't know if this will smooth things out or if it will be jagged, but roughly speaking, that's my guess for the top left. And I can do the same thing in the top right. Here, I use the knowledge that I've gained from all the papers that I've read. For example, we know that whenever people start out with a lower antibody response, they should end up with an even stronger response. This is an antibody ceiling effect. And so the lower you start out, the higher you expect to be. So slightly higher than what I'm guessing up here. And then the same thing in the two bottom left panels. And now I can let the model take its own guess at what's going to happen. It learned in 2016 what the vaccine responses looked like. It predicts in 2017 what it expects the post-vaccination responses to be. And so the model guesses pretty much the same thing that I do in this top left corner. In the top right, it didn't read the papers that I read, so it doesn't know about this antibody ceiling effect. So it actually hugs the post-vaccination response, the pre-vaccination response more closely and guesses that there's going to be a smaller response. And then the bottom cases were pretty much on par with one another, maybe slight differences in the jaggedness of the response. Okay, and now that you see what I think should happen, here's what they measured at day 21 post-vaccination in these individuals. So in the top left, where the model and I agreed, the data in fact agrees nicely with us as well. The chords that I'm showing you are the differences between the model prediction and the actual measurements. So we're aiming for as little of these vertical green chords as possible. And in the top right, whenever I guessed that the post-vaccination response would break high and the model guessed low, the response was in fact even lower than what the model guessed, which I found to be extremely shocking in that perhaps the model was latching onto features that we don't even appreciate yet. With this antibody ceiling effect, maybe there are caveats on it happens under these conditions, but not under other conditions. And then in the bottom cases, there are scenarios where we either extremely overshoot the predictions or undershoot the predictions. These happen in about 5% of cases and 10% of cases that we over or under. And in about 85% of cases, we predict spot on with what you expect to actually see. The error here shows you the error between our predicted values and the actual measurements. And because the assay itself has roughly a 2x error, then anything that's got a prediction error of around twofold should be as good as what you can possibly expect to have. We're essentially hitting the minimum in these cases, and then anything that goes above 2x, you can see that there's room for us to improve here, sometimes quite a bit. So 4x is considered now the bare threshold of what I'll tolerate as an acceptable prediction. Anything bigger than 4x like this is a complete loss, and we need to do better on that front. But nonetheless, if we have just these very basic examples where we're only using one data set for training and then testing on another study, we already see that there's the possibility here of using these past results in order to understand the heterogeneity that we see nowadays among different people. Because everyone starts off with their own unique pre-vaccination landscape. Can we understand what that looks like based on these types of models? The difficulty is that there are hundreds of different people in each of these studies. I'm only showing you 4 out of 300 in this case. So I'm going to switch views here and show the results in a slightly different form. Instead of showing all of the viruses for each person, I'll show just one virus for all people. So I'll take this leftmost virus and I'll show you the difference between the model prediction and the measurement. So this vertical chord here. So model prediction and the measurement, model prediction, the measurement, the difference between the green point and the green line. So that's what it looks like here for this 1968 virus, where we want for the values to be as close to this diagonal band as possible. And in general, you see that they are. Again, the error here being around twofold means complete victory for our end. And as we go to newer and newer viruses, we see that in general, we do a very nice job, although we do have still room for improvement. Some things are just flattening out here in ways that we don't want them to be. So this is where we'd like to do better. So how can we, and even in this scenario, I'm showing you here 6 viruses, which is all I can fit on the screen, out of our about 15 viruses. So here's what they all look like, just their prediction error at the end of the day, where something around twofold is considered to be a good result. These ones that are up here are between 3 and 4 are something that we should do better at. So across this, what we see is roughly 2.3-fold error on average across the 2 data sets. It's a very good start. Can we do better? All right, so here are the flu vaccines from the past 6 years. Let me show you all of the different vaccine studies that I use in a normal day-to-day life. And I take one of these studies and I ask, where can we predict out from it? This 1997 study here measured its own series of 30 different viruses. I ask, can any of these future vaccine studies, do they measure the same viruses, at least 4 of the same viruses that were measured here in 1997, so that we can predict how responses happen between one year and another? And it turns out that even though this was done in 1997, and even though time has moved on here, even a study that was done here in 2018 still measures enough of the same variants as this 1997 study that there's enough overlap to make predictions, which is pretty astonishing. It tells you how far people go back with their studies in terms of the variants whose responses they measure. And this is just looking at one of these studies. I can show you for every possible study, there is a cord that connects it to another study if there are enough overlapping variants to make any predictions whatsoever. And this just shows you that predictions are possible almost everywhere. It's nearly, I mean, it's a fully connected graph. There's one cluster here and one cluster to pretty much everything else. But among the scenario, we can predict between different vaccine studies that we see, but that doesn't necessarily mean that the predictions are accurate. So here's what it looks like if I add prediction accuracy on top, where I'm going to show this flashing back and forth a few times, and the chords that are becoming more transparent are the ones that are poorly predicted. So the ones that fade away are the ones that we predict poorly. The ones that remain are the ones that we predict accurately. And this is one of those scenarios where either whether 2 vaccine studies are accurate or whether they poorly predict, both scenarios are interesting. Because we want to be able to predict between 2 studies accurately. That's the ultimate goal. And yet, even when 2 vaccine studies cannot predict one another, that still tells you that there's something important to be learned in that case. So let's go and focus on those specifically. What led to one of these vaccine studies that leads to systematically poor predictions in other vaccine studies? What are the features or scenarios? Which people did they measure? What location was it at? What are the common traits that we see that lead to bad predictions overall? And what we find from these cases are that it's things like the time when the data set was made. This one is almost like a positive control. We expected it to be that if you have a measurement done in 1997 or 1998, and you try to cast that forward at least 10 years forward, we're going from 1998 to 2009, the predictions start off okay, and then they rapidly fall away as you get to older and older, to newer and newer viruses, as you're trying to project more and more forward in time. So you can predict some amount forward, but you can't go forward indefinitely because antibody responses are very different. We also find that the exposure history matters a lot. We had this very interesting data set here, number 11, where we looked at children who did not get infected or vaccinated in the past 5 years. And it turns out that those children couldn't predict any other data sets, not adults, not children, very accurately in general. And it doesn't mean that children are different from adults. We had other children studies that can predict adult data sets totally fine, but it's children that were not infected or vaccinated for 5 years that had these very weird phenotypes. And so exposure history matters a lot, perhaps in combination with age. And we also found that the type of vaccine that you receive also matters a lot. So right now, if you were to get the flu vaccine, there are about 7 different options. A lot of them are the shots that we get into our arm, but one of them is this flu mist nasal spray. And we know that this behaves in a very different way. It doesn't induce the same antibody responses, and we see that very clearly when we try to predict from one data set to the other. So this creates various buckets that we have that tell us when should we expect that studies act in similar ways. There's one bucket for when the studies were conducted, new versus old. There's another study for what was the exposure history of the people in the study. And then there's another bucket for which type of vaccine were people given. And so if you take all the studies that fall into the same bucket, such as all the current vaccine studies that were past 2009 and onwards, everybody who has an exposure history that was infected or vaccinated in the last 5 years, and if you look at everyone with the same vaccine type, like everyone who got the intramuscular shot, then we can restrict down to those specific studies and ask how well do they predict one another. And this is the case where now we're guaranteed that every study in here predicts the other studies, at least one of them, extremely well. And with this, we can ask now the final question of how far back should you go in time? If I'm going to predict here in 2023 what happens in the vaccine study, should I go back to 2022 or 2021 or even further in order to make the best predictions that I can? And we can literally do that explicitly. We can ask, as I go back n years in time, how do my predictions look? And the sweet spot appears to be at around 4 years, where you have the maximum predictions that you can make with the minimal error that you can make. So that's going to be what the final algorithm is that we currently use today. And we know, because we make 100,000 different predictions on it, we know that we expect on average 2.8-fold prediction error. And so that tells us that we have these robust prediction frameworks. We test this on about 1,000 different variants, flu variants that we have across these different studies, across different years, across different time points. And so this is the solid, robust, correct in 85% of the cases prediction framework that we use. But now with this algorithm, we can start to ask questions about what do the responses look like? What can we learn from them in terms of how to make the best vaccines possible? Okay, so this is where I'm gonna turn here into an interesting and curious facet of the antibody response, which is that as we look year after year on different individuals, we see that around 30% of people exhibit little to no vaccine response each year. It's as if they get the flu vaccine and we look at the response 1 month post-vaccination and they look almost identical to what they were pre-vaccination, as if they didn't get the vaccine at all. So what happens in these cases? Can we understand who these 30% are? What made them have a weak response? And so that's the next part of this talk. This is just going to be a snippet of what we're working on nowadays to understand the weak versus strong responses. I'll give a quick pause here to see if anyone has any questions thus far before moving onwards. All right. So the second part of the talk is going to be about how can you elicit a strong response? What are the features that lead to someone responding weakly versus strongly? So I'm going to show you here 2 examples. This is a weak response and a strong response, both of which we saw in the same year with the same vaccine. And these 2 individuals were different. You can see that their pre-vaccination landscapes, their day 0 gray curves, looked fundamentally different. This person started off very low across all of these different viruses, whereas this person right here had a nice peak here for this older strain, and then a little bump, and then they had a stronger response here for these newer strains. So their gray curves are different, and it makes sense then that their green post-vaccination curves will also be different. And indeed they were. This person here has this nice pyramid shape and then a little bump here in the newer strains. This person went all out, just the full hog, super strong response against these new viruses with these various bumps here for these older strains. And now you can ask, well, what is it about these responses that generated a strong versus a weak response? And so the way we can do that is we can go up and we can ask through our model, what do these responses look like? So I'm going to use the model predictions here, which are shown with these dark green points, as a proxy for what the actual post-vaccination landscape looks like. It's not a perfect representation. You can see that we're off according to these green lines, but roughly speaking, if you squint your eyes, the green points and the green lines look pretty similar. And the advantage to using the model is that we are now no longer limited to just the specific phenotypes that we've seen, just these 300 individuals that were in the data set. We can explore with the model what any possible pre-vaccination landscape would have looked like post-vaccination. So let's use the strong response as our target of where we want to go. I'm going to just phase out everything but where the model predictions land for post-vaccination responses. And I'm going to show you in dashed where the pre-vaccination day 0 responses were. So we know that this particular gray curve leads to this specific strong green response. And we can now overlay that on top with the weak response, where we have a different gray curve leading to these solid green lines, solid green points here. And we can ask, well, how do we push up the dark green points to the dashed green points? What does it take in order to make that? If we just had a single one of these features in the gray curve that was bumped up, could we shove up all the green points up simultaneously, or do we need to push in multiple different places in order to get them to match up? So that's literally what the model can do. We can say, well, let's nudge this pre-vaccination point up here, 1, 2, 3, take it up to whatever point we want. And then we can take another place and nudge it up and up. And we can keep going as far as we want, however many points we want to change this way. And then we can ask, well, what happened if we would have gone down a different track? What if we start over and we say, let's nudge this point up instead to a different amount? And then we can try maybe taking these older viruses right here and nudging up their responses. And in this way, what we found so far is that there is no global master switch where you nudge up this and that point and all of a sudden all the green points shift up. It all appears to be pretty locally contained so that every change that you make to a specific virus nudges it up, but not so much the viruses around it. So not as much cross-reactivity as one might have imagined from the vaccine strain bumping you up. You essentially bump up different values, but they all bump up individually based mostly on where the pre-vaccination landscape looked like. And this is where we're now adding features such as, what happens when we add age into this profile, or a person's genetics, or HLA type, in order to predict specifically how they will respond. And is that what is going to end up distinguishing the many strong responses that we've seen from these weak responses that we have plenty of as well? All right. So that's how we're looking at these types of responses. And now I'm going to shift gears one last time to give you the future and where we're going with this in order to understand, if we know how people will respond to different vaccines, then can we predict which vaccine is going to be optimal for them? All right, so this is the part that I'm doing here with the Prepare Institute. The idea is that about 30% of people have a relatively weak post-vaccination response, which I'm defining precisely here as an HAI of less than or equal to 40 against the vaccine strain. So let me show you an example of this. Here's a person who starts off, this is all real data here at the bottom. It's a person who starts off with this gray curve as always, and whenever she gets the vaccine, it bumps up to this green curve that's only slightly larger. Against the vaccine strain itself, she ends up less than this HAI equals to 40 line. And so this is considered to be a weak response here. And if you follow this person year after year, what you find is that just because you're weak in one year does not mean that you'll be weak in other years. In fact, what we find is that around half of the people who are weak in any given year tend to be strong either in the preceding year or the subsequent year. So weak responders aren't weak because they're genetically predisposed to be always weak. Rather, they are weak because something about their specific antibody response and that year's specific vaccine just did not work out. And in a different year, they might have had a much stronger response because that year's vaccine meshed a lot better with their specific profile. So here's an example of another person who, again, they look like they have a weak response, but don't give up on them because in 2022, out of nowhere, they had a very strong response. Here's a third case where this person just couldn't make up their mind. They started off weak, they went to strong, they started off weak, then they went to strong. And again, we don't know what is causing these specific profiles to happen. So our question is, if we could predict that in 2021, this person were to have a weak vaccine response, could we then go up to them and say, all right, most people are indeed getting this 2021 vaccine this year, but we think that you would actually benefit from getting last year's vaccine response once again, because we know that you react very strongly to that one. Then could there be a subset of individuals who would actually benefit not just from getting the most up-to-date vaccine, as ironic as that sounds, but actually from getting the vaccine that is best suited for their specific antibody profile? And so this is the prediction challenge that we're doing right now. We're trying to understand, are there people who would benefit from getting last year's vaccine again? And it's a very nice question because we don't have to recreate a new vaccine from scratch, which would take a lot of work in clinical studies and safety measures and all that. Rather, we take the previous year's flu vaccine, which has already passed all health checks and people have received it and we know that it works. And we ask, are there people this year in the fall of 2024 who should actually get the 2023 vaccine again? So just to show this pictorially, we do this with Ted Ross here in the Cleveland Clinic off in Florida. He has gone in 2020 and given hundreds of people the 2020 vaccine and then measured their responses pre and post vaccination. And in 2021, he did the same thing, giving people the 2021 vaccine, 2022 vaccine, and the 2023 vaccine. These two happen to be the same ones because the H3N2 vaccine did not change. And now in 2024, I've asked Ted if he will do the same thing he normally does, give 200 folks the 2024 vaccine, but now give another 100 of them last year's vaccine, the 2023 one. And we're doing this with a small cohort here at LGI with our clinical core. Ted's doing it with a larger cohort there at the Cleveland Clinic, just to make sure that geographic locations don't have an effect. And the question is going to be that, can we predict the responses from both of these types of individuals, the ones receiving the 2024 vaccine and those receiving the 2023 vaccine? Because if we can do that, and we can say you all received vaccine A and the other group that received vaccine B, and we can predict what happens in both of these cases, then we can do computationally what no experiment will ever be able to do, which is to take a single person and predict what would happen if they received vaccine A rather than vaccine B. Because experimentally, as soon as you give somebody a vaccine, you can't undo it, give them another vaccine. Instead, but computationally, you can do that. Using their pre-vaccination profile, if you can forecast what happens, then you own the system. And just as a fun side note here, I originally pitched this to Ted as, let's go ahead and optimize the vaccines here. We'll use our prediction profile and we'll tell each of these 300 people, I think you should get vaccine A versus vaccine B based on their initial pre-vaccination landscape. But then Bjorn pointed out that it's actually better to do the much simpler study of just assign them all randomly, because we want for some people to have these strong responses versus weak responses in each of these categories, those receiving the 2024 and those receiving the 2023 vaccines, so we can show that our model can predict the full dynamic range of responses. And if we can do that, then go ahead and the following year, try to optimize it and predict it for everyone. So this is very much what we're doing. It's what is being collected now in June for the 2023 recipients, and it's what will be collected in the fall for the 2024 vaccine recipients. And we are going to see what the model can do and what it can predict. At the moment, we have this challenge of not being able to predict the differences between these two vaccines very well. We can take somebody who was there in 2021 and 2022 with almost the same pre-vaccination landscape, but our predictions for what the vaccine response looks like in each of these cases tends to be very good in one of these years, but not always great in another one. So this is what we're doing computationally in the lab, is making these types of predictions better. We have our timeline, which is by the time that Ted makes his experiments at the end of this year, we need to be able to do this much better. We have all the data in order to do that. And so what we're looking at is how do we make these types of more nuanced predictions even better than what we have right now. And we're specifically looking at how you can encode the viral HA sequences into the vaccine predictions, because we know that some years the vaccine changes just a little bit, in other years it changes quite a lot. We should be able to incorporate that in to say that our vaccine predictions are very similar in these kind of scenarios and very different here whenever the vaccine strain changes. And we're also looking at many other aspects of the immune system, not just the pure antibody response, but also T cells, B cells, and genetics. And of course, we're trying to predict not just what happens at the 1 month post-vaccination time point, but also at 3 months, 6 months, and 1 year to understand not just how strong the response is, but also how long it lasts. All right, and with that, I'm going to stop there, and I'm happy to take any questions and even to hear how these kind of scenarios might fit into what you all do at the Prepare Institute. Thank you very much.
Yeah, thank you. That's fascinating. When are you going to know your results of your clinical studies, or have an estimated time?
So they're going to be collected in, they're going to be collected in September, which means we'll know by December what their results will be.
And I might have missed it, but how are you adjusting or collecting whether or not they've been, been exposed or got seasonal flu? Or
okay, that's a great question, and the answer is that is probably one of the hardest things to measure, because self-reporting is terrible, and experimental measurements, you have to basically track people over time very closely. So nobody knows how to do that. The one advantage that we have over other people is that this information on exposure history is embedded within the landscapes that we look at. So those gray landscapes that look at how do you respond to this year's virus and last year's virus and the year before that, others have shown that those peaks and troughs show you when you were infected last. Essentially, every peak represents an infection. It's a bit more complicated, but what we think is happening is that our model, which we never give it at the moment infection history implicitly, we don't even give it age information, it takes in those data, does whatever calculations it needs to do to figure out what the exposure history is, and then uses that whenever it makes the predictions. So that's how we're at the moment dealing with it very indirectly, but it's a great idea to actually incorporate that directly through the previous work that has been done. Great question. There's something in the chat. Okay. So Stephen's question is, how can these studies reduce the need for multiple large clinical trials of vaccines? This is, okay, it's a good question, and I am not in the clinical space, although I would very much like to be in there and help contribute. Where I think we can help a lot is that you have to make many decisions when you create a vaccine. everything from the dosing that you give to how far apart will those doses be, to do you add an adjuvant, or do you create the antigen in this way or the other way so that it can best be presented to the immune system. And what we do is we take those vast number of possibilities and we sift among them, and we say, we think that these 5 methods should be prioritized out of these 5,000 that you can do. And so that's really where it's about learning what people are doing. It's about learning how we can help. And I think that whenever people design their new therapies, that it's very powerful to go back and ask, what can we glean from past studies that can help inform our efforts? And not just, I've read these papers and I've made my stand and I'm going to do it in this way, but really like letting those past works predict in the future on what we think will happen if you choose route A versus B versus C versus D, and on and on and on. I think there's real potential in doing these types of analyses that is being underutilized at the moment. And there's a hand raised.
Yeah, that's me. Oh, Mary Garrett Ovania. So I know that a lot of the vaccine and your program, your model deals with viruses that occur year after year, and the vaccines are made for that. And you said something about that the response is not, it has nothing to do with the genetics of the patient or the person. And so my question is this, I think you said it has to do with the vaccine and the response, the cytokine response to that vaccine. So my question is, in situations where there's no continuous infections and so on, let's say you go in, the person has not been exposed to anything, let's say it's a bacteria, right? Mm-hmm. And so maybe you look at the immune, you just run cytokine inflammation, and then from there, can you predict that this vaccine would work on that person, given that this is their cytokine profile? If you gave them this vaccine, how are they going to respond so that you select which vaccine to go with?
Okay, good question. So what I mentioned about genetics is that we aren't using it yet, but we're definitely interested in using it. I think that HLA type will absolutely matter. And we're also looking at cytokine profile. We're also looking at T-cell responses. That's all happening towards 2025. In terms of your question, I think that I don't know the answer to whether it will matter or not to look at cytokine profiles, but if there are existing studies that have basically looked at a person's cytokine profile and then looked at the post-vaccination response and associated the two, then we should be able to learn for new individuals, if you measure their cytokine profile, how will they react to that same vaccine or treatment or immune perturbation? And so I mean, the power here is that anybody in the world could have done these studies at any time, and then you get to reap the rewards of that information. Does that address the question?
A little bit, in that I think you're saying you would need the information for pre and post vaccination. But I was just thinking when maybe it's just, there's no post vaccination results, but you're looking at somebody, you want to start vaccinating them, and maybe it's a one-time vaccine versus the viruses that have to be, the vaccines are given every year. So there are certain vaccines you give maybe once and maybe a boost after 10 years, but you're trying to determine which is the best vaccine. So you won't have the post-vaccination information. I wonder whether you can use your model to predict which vaccine to use for somebody when you're vaccinating people who have never been exposed and so on.
You certainly will need some information about some outcomes, even if there's not like a post-vaccine, like whatever you're trying to measure is your metric of success. So you'll take these people, you give them vaccine A, and you'll measure something from them 1 month, 10 months, a year, whatever it is down the line, and then you'll give them vaccine B and you'll measure that same thing. We need essentially a starting point of enough people who have gotten those types of measurements, and then from there we can infer what will happen in new cases. We can't work from scratch yet, but whatever you're measuring should be enough to predict new cases. So either you're the first person who's ever measuring these at all in the world. And then we can use a part of your results to predict what will happen when you do it in the future to new people, or somebody else somewhere has measured it already, and we can use their results to predict what will happen in your studies.
Thank you.
Happy to talk more on that front.